The preoptic area, or the POA, is the portion of the hypothalamus in the brain responsible for regulating reproductive physiology and behavior. During sexual development, the POA is completely restructured. Mammalian gonads are also completely restructured during development, including maturation of eggs and sperm and release of sexual hormones. Rat masculinization occurs following spikes in male sex hormones, known as androgens. Because estrogens, female reproductive hormones, are not secreted during development in the same way, mammalian feminization was long believed to be a passive process. This idea is now being challenged, but the signal to begin development is not yet known. The concept of mammalian feminization is a typical chicken or the egg type problem. Does the brain tell the gonad to begin sexual development, or does the gonad tell the brain? Here, Dodd et al. 2019 explore this concept using sequential hermaphrodite clownfish. Clownfish form mating pairs, with the female being the largest and most dominant in the pair. When two male clownfish form a new pair, dominance is established amongst the mature males. The dominant male will then undergo feminization and transform into a female. If an existing pair loses its female, then the mature male will transform into the dominant female, and one of the undifferentiated or immature fish will mature into a male. Because clownfish development is easily manipulated, they make excellent models for developmental biology. In a laboratory, 34 mature clownfish males were removed from their female partners and randomly paired up. Pairs were observed for the first hour after pairing, where individuals approached each other and began the battle for dominance. They were then observed every day until dominance had been established, which took a few days. Once there was a clear hierarchical structure, pairs were sampled at 3 weeks, 6 months, 1 year, and 2 years for hormones in the blood. Brain and gonad histology were also examined at these times. Over the 2 years, the dominant fish grew, while the subordinate one did not, leading to a visible differentiation in morphology. The brain of the dominant fish began feminizing with an increase in the number of POA cells. Gonadal tissue degenerated in both members of the pair, possibly to divert energetic costs while reproduction was not necessary. In clownfish, mature males have the reproductive organs of both sexes. They are ambisexual. Testicular tissue makes up approximately 70% of the gonad, while non-viable eggs make up approximately 30%. Throughout feminization, testicular tissue continues to degenerate until none is left. The final step to a successful sex change is when the gonad fills with viable eggs. Despite plenty of evidence of sex change occurring, only 3 of the 17 pairs completed gonadal sex change within the time frame of this study. In this study, feminization of the POA occurred first. Male gonad tissue began degenerating early on but did not appear to be directly related to the sex change. Female gonad tissue did not differentiate until much later, and sexual hormones did not change until even after this. In other words, clownfish brains undergo sex change before the gonad, sometimes even years before. It appears that sex change begins in the brain, and the sex organs can remain ambisexual until some yet unknown signal conveys the message to complete the process. If these results hold true for mammals, we might expect female development to begin in the brain, possibly years before changes in sex organs. These results may not directly apply to mammals, but they give the first evidence of active feminization. They are an interesting story, and they are a good place to start with future work. This post was originally made on August 22, 2019 by Brandy Bagar. It was titled Sex Change 101. It Starts in the Brain. It featured a paper by Dodd et al. 2019 titled Active Feminization of the Preoptic Area Occurs Independently of the Gonads in Amphipriorian Ocellaris. This has been Oceanbites Out Loud. Check out oceanbites.org for this post and other great posts. Thanks for listening.